Hello, and thank you for joining us. I'm Gwen Taylor, Senior Developmental Editor with Current Protocols at John Wiley & Sons, and I'm delighted to introduce today's webinar titled Cancer Stem Cells, Hypoxia, and Tumor Treatment Response. This webinar is being co-sponsored by Current Protocols and Baker Ruskin. Baker Ruskin is the trusted source for controlled atmosphere solutions for tissue and cell culture applications. Its advanced line of hypoxia workstations and media deoxygenation solutions help improve reliability and reproducibility and achieve more accurate results by precisely mimicking in vivo physiology in the atmosphere and in culture media. Current Protocols is in its 28th year and is the largest collection of peer-reviewed, authoritative, and regularly updated step-by-step research protocols available for life scientists worldwide. With 17 titles and over 17,000 protocols, Current Protocols is part of Wiley Publishers. During today's program, we encourage you to submit your questions throughout the event by clicking on the Ask a Question box at the bottom of your screen. Your questions will not be seen by any of the other attendees. The webinar will be recorded and available for viewing in the next few days. We will send you an email with details of how to access the on-demand webinar, along with a customizable certificate of attendance and a PDF of the slides. So now it is my pleasure to introduce today's speaker. Dr. Richard Hill is a member of the senior scientific staff at Ontario Cancer Institute, Princess Margaret Cancer Centre, which is part of the University Health Network in Toronto, Canada. He is also a professor in the departments of medical biophysics and radiation oncology at the University of Toronto. Dr. Hill's research program focuses on laboratory and translational research studies in tumor and normal tissue radiobiology. Over the past 25 years, he has been actively involved in studies of hypoxia in tumors, focusing on issues associated with metastasis and stem cells. His lab published initial work on stem cells in tumors relating transplantability to treatment response more than 20 years ago. His lab was also the first to demonstrate that exposure of cancer cells in vitro to hypoxia increased their metastatic potential when injected into mice. Further work demonstrated that hypoxic levels in animal tumors in vivo also predicted for metastasis. These findings led to studies of hypoxia in cervix cancers and the development of orthotopic xenograft models of human cervix cancers. These models have subsequently been used to examine the proportions of potential stem cells in hypoxic regions of these xenografts. Dr. Hill has published over 300 scientific papers and has received numerous awards, including the Robert L. Noble Award by the Canadian Cancer Society for Excellence in Cancer Research, and the H.S. Kaplan Distinguished Scientist Award from the International Association of Radiation Research. He is also co-editor of the Basic Science of Oncology, a book that is widely used in basic courses for oncology trainees. So let's go ahead and get started with a very warm welcome to you, Dr. Hill.
Hello, and good day from Toronto. What I'm going to talk to you about, as you've heard, is the concept of stem cells and work that we have been doing, really looking at the role of hypoxia in the phenotype of stem cells, and also we're going to talk a little bit about the cervix cancer model that we have set up, that we have been using to investigate some of these issues. And finally, we're going to talk a little bit about issues associated with the tumor treatment response in the context of stem cells. The focus of that work is going to be primarily on radiation, since we don't really have time to deal with the drugs as well. So initially then, the question is, what is the concept of stem cells? Many of you, I'm sure, are familiar with this concept that the stem cells sit at the top of a hierarchy of cells, which are generally called progenitor cells or transit amplifying cells, and they create then the final post-mitotic cells in the tissue. And the specific feature of stem cells, which is important, is that they are self-renewing, this being then the population of cells that can regenerate itself. I'm just want to just make a note to you here that in the literature you will find various different terminologies for this cell population. Cancer stem cells, CSCs, cancer-initiating cells, CICs, or tumor-initiating cells. All of these are really essentially the same cell from the point of view of this particular issue. So the data on stem cells, or the fact that not all cells in the tumor really were capable of regrowing that tumor, is quite old. But the most recent interest in stem cells really arises from work from about 10 years ago, where people were actually working with different markers on the surface of cells to actually see if they could actually sort cell populations which would actually have increased stem cell ability. So the initial work was done really using breast cancers, where they looked at these markers, the CD44 positive, CD24 minus or low marker really, and they showed that if they sorted the cells by flow cytometry, these cells had increased ability to transplant the tumor. And as you can see from this slide, there are a very large number of different markers which have subsequently been identified, which seem to enable one to enrich populations for a stem cell phenotype. And it may also be important to recognize that a number of these surface markers may also be expressed on normal tissue stem cells, so they're not necessarily specific to tumor cells. So, as I said then, the way this has often been done, usually been done, and the gold standard for doing this is really to test the stemness of a cancer cell, is to transplant it into normally, this is normally done with human tumors, cells transplanted into immune-deprived mice and recently. And what is done is you look for the fraction of cells in the population which are required to actually have a tumor regrow. There's also possibilities to do this in vitro using what are called sphere-forming assays, and these are often reasonable substitutes for the in vivo assays. They correlate reasonably well. And you need special media really to do this, but we don't need to go into that. There are a number of caveats, though, particularly in relation to various conditions which can impact on the ability of the cells to actually form tumors. Culture condition, the animal model can be important, and in some cases people have added various feeder cells, and one of the common things which is added very frequently is a gelatinous substance which is called matrigel. It's generated from a specific cell type, and it contains various proteins and also growth factors which may impact on the ability of the cells to actually grow. So if we look then at data for colon cancers, this is data published a number of years ago by Catherine O'Brien, showing really that what they did here was they sorted the cells based on the expression of the surface marker CD133, and then they injected different numbers of cells into the tumor. Into immune-deprived animals to find out how many were required to actually form a tumor. And you can see here that the number of cells, this is A, the number of cells which were required from the CD133 positive population was of the order of about 200, whereas the CD133 negative population, that number was more like about 700,000, so it's a very large number. And in fact, these small number of tumors that they actually saw, is possible that they actually represent contamination from CD133 positive cells, because the sorting procedure is not perfect. If you don't sort at all, and you just put the bulk population in, you see that the number that they required to get tumor takes was about 60,000. Now, this is an average from a number of different tumors, 17 tumors that they studied, and the data was put together really just to demonstrate the concept that there are these kinds of differences that can actually be seen. And there are a number of different data sets which have looked at that. So here's another data set. This is from Quintana, looking at melanoma. Now, they didn't sort their cells. What they did was they looked initially at different types of animals. These are the non-SCID animals are immune deprived, but if you knock out the gamma T null mutate, give them a gamma T null mutation, they actually become more immune suppressed. And what they were able to show was that for this particular study in the human melanomas, there was actually a greater chance for the cells to form tumors if they were injected into the so-called NSG mice. They also showed, again, and this was what comes back to what we were talking about before, that if you actually inject the cells with Matrigel, you can actually get a higher take rate. And the overall data from this particular study was quite interesting in the sense that they showed that in their study that an average of 1 in 4 tumor cells was a stem cells from the human melanoma cell lines that they looked at. And that, of course, is substantially larger that you recognize than the 1 in 200 that was shown in the colon. So there's a general thought that what one is doing here is actually enriching for stem cells rather than just selecting only stem cells. The other issue is that it isn't necessarily specific to the sorted population. So here's data for ovarian cancers, where again they use CD133 as a marker, and you see a big difference in the frequency of tumor-initiating cells between the CD133 and the plus and the CD133 minus population. But you do see situations where the CD133 minus population is actually inducing tumors. Now, in this particular study, they actually looked at the question of whether there was possible contamination of the CD133 negative population by CD133 positive cells. And in some cases, they could detect that. But in other cases, they were able to rule out that was the explanation for why they were seeing tumors from the CD133 negative population. So it's clear that while we're enriching for stem cells in the population in this particular experiment, there are cells which are not expressing this particular marker, which are also have a stem cell phenotype. And we'll actually see that later as we go forward. Now, as I said to you earlier, there's a lot of old data in the literature also on showing really that different tumors require different numbers of cells really to actually induce a transplant. And this is data taken from a paper by Hewett, which is from about 40 years ago, where he actually put together all the data for studies that he had done on spontaneous mouse tumors. They're not mammary tumors, they're mouse tumors, and in a number of different situations. And he demonstrated that all these tumors essentially were non-immunogenic in the system that he was working in. So it's a nice study in the sense, and what it shows is that the number of cells required to get 50% takes, the so-called TD50 value, is quite low for leukemias, but it can be much larger for solid tumors, and it varies quite a lot. And one of the things that he did in this population, in this experiment, was he actually didn't add Matrigel, because Matrigel didn't exist then, but he did add large numbers of heavily irradiated cells to the populations of viable cells that he was actually injecting. So they gave the same tumor cells a big dose of radiation to kill them all, and then they mixed them with the viable cells. And that actually makes a significant difference to the take rate. Again, it's consistent with what we were talking about earlier, and in fact, the different aspects of the way you do the experiment can affect the result. And going forward, this is an experiment that we had done a number of years ago, in which we actually did this test where we actually estimated the number of stem cells by calculating the TD50 value, and that's plotted along the x-axis here. And we actually took tumors and we irradiated them with different doses of radiation to actually determine the dose of radiation, single dose of radiation that was required to actually cure those tumors. And this was done under hypoxic conditions to make sure that there was a uniform level of hypoxia in the tumors. And the hypoxia was induced just 1 or 2 minutes before the radiation was given. And what you can see is that there's actually quite a good correlation between the stem cell proportion and the dose of radiation required. So if you need very few cells to transplant the tumor, meaning that virtually all the cells are stem cells, then you need a large dose of radiation. If you need a lot of cells to transplant the tumor, meaning a relatively small fraction of them are stem cells, then you need a smaller dose of radiation. And the slope of that line is actually consistent with the expected radiosensitivity of hypoxic cells. And similar data from Dr. Milas's lab in the MD Anderson Hospital in Houston, where he split his data into data for carcinomas and sarcomas, and there's a slightly different slope to the line, but essentially you see the same result, indicating that the radiation sensitivity of these cell populations is in fact correlated with the stem cell proportion. This is the data set which I put in. It's actually for human tumors, and it was an experiment that was done many years ago. Probably I think would be regarded as non-ethical these days, in which they actually took tumors from patients and actually prepared cell suspensions, and they actually injected those cells back subcutaneously into the patient. So this was done to really just look at the question of how many cells would be required to transplant the tumor. And you can see again that there are very large numbers of cells required to actually transplant these tumors, suggesting that, again, that only a small fraction of the cells may have stem cell ability. It's important to recognize that in this experiment they only waited a total of 8 weeks in some of the studies. So this is a very short time window and probably isn't long enough to be certain that the tumors wouldn't grow with some of these smaller numbers. But again, the numbers, as you see, are quite high. Southern actually thought that this was probably an evidence that tumor cells were immunogenic in the patient, and that is interesting in the current concept of the studies which are going on with immunogenicity. So one of the issues which has come out of the studies of stem cells really is the concept that the cells may sit in a kind of a niche environment, and work from Jeremy Rich's lab has really identified potentially two types of niches. There's a so-called perivascular niche, where the cells may sit in an environment, very close to vessels, or there's a hypoxic niche where the cells sit at a significant distance from vessels, and their data had suggested that these may be preferential sites for stem cells to survive. And this is consistent with work from Dr. Wieke's lab, where he has in fact looked at stem cells using the marker aldehyde dehydrogenase, and what he looked at was the situations of how these cells were in relation to hypoxic regions in breast cancers, and they actually induced hypoxia by treatment of the tumor-bearing animals with sunitinib, which is a drug which actually blocks angiogenesis. So what they showed here was that here's a measure of hypoxia using a probe for hypoxia called Hypoxia probe, showing that in the sunitinib-treated animals there are significant regions of hypoxia, and you see here is the marker, aldehyde dehydrogenase marker, showing that there's a much higher proportion of cells expressing this marker in the hypoxic region. And if you look over here, you can see here's the quantitation, just showing the percent of cells which are expressing the aldehyde dehydrogenase marker. And this is much greater in the hypoxic region. And this is another example of a situation where hypoxia may in fact enhance the expression of the stem cell phenotype. This is for medulloblastoma cells. This is a cell line, and they grew these cells at different oxygen concentrations, either at 20% oxygen, that's air essentially, or at 2% oxygen. And what they showed was that the percent of the cells which were expressing the CD133 positive marker was higher if they grew the cells at low oxygen levels. They also showed in this experiment that there was a slight difference in the radiosensitivity of cells expressing the CD133 marker versus cells that weren't expressing it. That's a small difference, and we'll come back to talk a little bit more about that later as we go forward. Another quite interesting aspect of the stem cell phenotype is the concept that it may also be associated with the so-called EMT, or the epithelial to mesenchymal transition phenotype, which has been associated with metastasis. And so here, again, with the mammary tumor model, they're using CD44 high, CD24 low as the marker of stem cells, and they're looking at features of those cells which relate to the EMT transition. So cells which have not undergone EMT tend to have high E-cadherin. These are epithelial cell populations, so they have high E-cadherin and low fibronectin vimentin. If they've undergone this transition, then they tend to lose E-cadherin expression, and they have upregulation of fibronectin and vimentin. And what was shown in this data set, which was quite interesting, was that they were also able to see upregulation in the messenger RNA of various populations which relate to genes which are actually involved in, believed to be involved in the transition of the cells into the EMT-like phenotype. And the ones which are of particular interest to us are these genes at the end here, twist and snail, which is now called SNI1, which are hypoxia responsive genes. So expression of increase in hypoxia will actually upregulate those genes. So here's another data set which shows something very similar to what we were talking about before. Here they're looking at two different markers, in fact, for stem cells in mammary tumors. Here is the CD24 low, CD44 high marker, and here is the aldehyde dehydrogenase marker. And by and large, what they're seeing in these populations is that these markers are on different cell populations, suggesting that there may be more than one population which has stem cell features in the tumor. And there's a slight overlap here, but it's very small. What is interesting about this data set is that what they did was they actually sorted the cells based on these markers, and then they grew them in culture for a period of time, for 10 days. Either the bulk population or the sorted populations. And what they showed was that here, as you can see, that this is the aldehyde dehydrogenase population, which was sorted initially. There's still a significant fraction of cells which are showing this marker, but there's now a very large number of cells which are not showing the marker, which are just like the bulk population. And the same is true for the CD24 minus CD44 high marker. Again, you see significant fraction of cells expressing this marker, but a very large fraction of cells have actually lost the expression of that marker. And that was true for the two different cell populations that they looked at. And it's consistent with the concept that cells can move backwards and forwards between an EMT-like phenotype and a MET-like phenotype, and they associated these with the aldehyde dehydrogenase marker and the CD24, CD44 marker. So this is an interesting concept that, in fact, there may be the ability of cells to be plastic and move backwards and forwards. So this can be kind of summarized then in this concept where we have the development of a tumor which contains a certain fraction of stem cells, illustrated in yellow here, and that these cells, if they're exposed to hypoxia, may in fact be able to transit into an EMT-like phenotype, and genes that can be involved here, SNI1, as I told you, snail, and this is sometimes called slug, SNI2, and this twist, and also TGF-beta can also be upregulated under hypoxic conditions and can do this. Interesting, radiation can also upregulate TGF-beta, and we'll come back to that as we go forward. And the hypoxia may, as we said, be able to maintain the stem cell phenotype in a kind of a niche. And these things all may relate to metastasis. So just moving then to talk briefly about the orthotopic model of cervix cancer that we have been developing. What we're seeing here is the growth of the ME180 tumor cells. This is a human tumor cervix cancer cell line, and what what we're seeing is the growth of these tumors actually in the cervix of the mice. So this has been transplanted with a small piece of tumor onto the cervix of the mouse and is growing there, and you can see here the DS-red expression. If you take that tumor out, what you see, in fact, is that this tumor has metastasized to lymph nodes up the periaortic chain here, and you can see these are marked by the DS-red marker as well. And we see for this particular tumor that there's also some evidence for tumor spreading to the lungs. So we have developed a number of xenografts, PDX models, directly using this technology, where we've transplanted pieces of tumors directly from patients into the cervix of mice. These are either NOD-SCID or NSG mice, and that allowed them to grow up. And what we've done is we've looked at some of the features of the microenvironment of those, particularly in relation to those tumors, using staining technologies, and then quantifying the fraction of stained cells in the tumors, and we've compared that against the actual fraction of stained cells in the original patient biopsy. So here, along the x-axis, is the fraction of the staining in the patient biopsy, and on the y-axis is the fraction of staining in the third passage of the xenograft. And there's a number of different markers that we've used here. What I'm showing you here is a cytokeratin marker. This is a marker which marks the epithelial cell, the tumor cell population, essentially. And what you can see in general is that there's a very good correlation between the fraction of cells expressing the cytokeratin marker in the biopsy and in the xenograft. And this ICC, the interclass correlation coefficient, is actually showing that you've got a good correlation here. Values above 0.8 are regarded as a good correlation. And there was one tumor that didn't correlate so well, but all of the rest showed a very nice correlation. And moving on then to ask the question about hypoxia, we see that the markers for hypoxia, we're using here CA9 as a marker for hypoxia. This is a gene which is upregulated under hypoxic conditions in tumors. It's also affected by low pH. And the red circles here represent CA9 expression in the tumor cell population, and the green circles represent CA9 expression in the stromal cells of the tumor. And you can see that, again, that there's a very good correlation between these, the biopsy and the original patient xenograft, and patient biopsy. And what you also can see, in fact, is that the hypoxia tends to be much more extensive in the tumor cell population than in the stromal population. So we've actually used the CA9 marker really to look at the issue of whether the cancer-initiating cells or cancer stem cells in the tumors may be related to the environment that they're growing in. And I'm showing you here initially an experiment where we just didn't do any sorting, but we looked at the number of cells required to give tumors, either from a number of these xenografts. There's 7, I think, xenografts here, and we actually injected either into NOD-SCID or NSG mice to see if there was any big difference between the two. And here we're plotting the frequency of the cancer-initiating cells, and the lower the number here, the more cells the less cells are required to actually get a tumor take. And you can see for some of the tumors, we needed less cells if we'd injected them into the NSG model. But in many of the tumors, there was a number of the tumors was really no difference. And if you look at the data here, there's a substantial overlap here. So we didn't think that was really a big effect. So we went forward primarily studying using NOD-SCID mice in our study, where we actually sorted the cells based on CA9 expression. So here we see high CA9 expression or low CA9 expression. These cells are the mouse cell population within the tumors. Those are discarded. And then the populations are sorted according to CA9 positive or CA9 negative. So here then is the data again, showing in the same way. We're plotting the cancer-initiating cell frequency here for the different models for either the CA9 positive cells in the open bars and the CA9 negative cells in the black bars. And you can see again that the CA9 positive cells, you need less cells to get a tumor take. In 6 of these models, actually significantly less cells, and in one of them we got some very similar values. And you can see, in fact, because this is a log scale here, that there's about a factor of 10 difference. So the majority of the stem cells in many of these tumors actually sit in the hypoxic regions of those tumors, but not all of the cells do. And so again, we're seeing a situation where there may be 2 populations of cells which have stem cell properties in the tumors. This data is actually consistent with data which has been published for the 41 breast cancer cell growing. These cells were growing as mammospheres, which, as I told you earlier, can enhance the fraction of stem cells. They're looking again at CD44 positive, CD24 low as the marker for stem cells. And what they've done here is they've grown the tumors and the cells under normoxic or hypoxic conditions, but they've actually transfected into the cells an shRNA to actually knock down CA9 in this population, or a non-specific shRNA in this one. And you can see that if you knock down CA9, the fraction of the stem cell population is very much increased under hypoxic conditions, but much less so if you don't have CA9 expression. So if we summarize that, that data, then what we've seen is that hypoxia can affect the expression of stem cell regulated genes, and I didn't show you the data here, but there is evidence that OCT4, which is one of the stem cell genes, is upregulated under hypoxic conditions. There's also evidence that epigenetic control of the regulation of certain genes can be affected by hypoxia. It's been shown that dicer, which is one of the important enzymes in the control of microRNAs, is actually downregulated by hypoxic conditions. And there's also evidence that methylation can be changed in hypoxic environments to enhance stem cell phenotypes. There's also data suggesting that hypoxia can actually enhance the ability of cells to become induced pluripotent stem cells, which is another sort of suggestion that hypoxia can drive the stem cell phenotype. As we've said, there's this niche environment for hypoxia, and there's also an issue associated with the epithelial to mesenchymal transition, which may relate to stemness.
Okay, now before we leave the issue of hypoxia, I just wanted to mention the fact that hypoxia is often indicated in the literature as though it's a plus or minus issue, whereas that, of course, is not correct. And oxygen concentrations vary over a wide range, and the way cells respond to different oxygen concentrations also varies according to the oxygen concentration. On this graph, we're seeing oxygen concentration along the bottom axis and response up the vertical axis. The red line here, the solid red line, is indicative of the expression of the HIF1 turn-on of the HIF1 growth transcription factor. And this relates, of course, to the turning on of genes like CA9, which we talked about earlier. On the other hand, the green line, which is further over to the left, indicates the uptake of the hypoxia marker EF5. And we can anticipate that hypoxia probe, which I showed you earlier, is probably in the same order of oxygen concentration. And you can see there's about a factor of 10 difference in the oxygen concentrations required for those two situations. So this is quite important. In the context of radiation sensitivity, you can see the black line, which is plotted in the other plane, shows how oxygen concentration affects radiation sensitivity. To the left, the cells are more resistant to radiation than they are to the right. And so you can see that if one is using a marker like CA9, one may see an intermediate level of protection from radiation, whereas a marker like EF5 is going to give you almost the cells which have completely protected. So it's important, I think, to recognize that there are these differences. And these lines, of course, are also dependent a little bit on the cell concentration of various cell factors in the cells themselves. For example, the radiation sensitivity line is actually dependent on the level of free sulfhydryls in the cell, since oxygen is actually competing with free sulfhydryls to actually react with the radicals on the DNA. So these lines may move a little bit depending on the exact conditions inside the cell, but they do illustrate an important issue that cells respond differently to different oxygen concentrations.
So, having talked about that, then it's important to recognize that there are actually quite a number of unresolved issues as we move forward in understanding stem cells. Probably the most important one is the issue of plasticity. What is the real level of the ability of the cells which are in the transit amplifying population or in the progenitor population to moving back into the stem cell population? There's a potential role of genetic instability in that context, which is really not well understood at the present time. And then there are issues associated whether treatment factors can actually influence the progenitor or transit amplifying populations, really, in relation to stem cell properties. So there's some evidence, and I'll show you this data in a minute, that as we saw, that radiation actually can, and also drug treatment, can actually push the cells back into a stem cell phenotype. And following on from that, the question is whether that might also change their treatment resistance, because there's some evidence that stem cells may in fact be slightly more resistant to treatment than the progenitor cell populations. And we're going to see that as we go forward. So we can think about this then in this context, where we've got going back to our original model, that instead of the arrows only going in one direction, these arrows can also go back, and the cells which are in these progenitor and transit amplifying stages may in fact be able to move back up this pathway. And that might be due to natural plasticity. As we've shown, it can be potentially due to tumor microenvironment. It may be related to genetic and epigenetic instability of the cancer cells, and it may also be an effect associated with the treatment. So this also relates actually to the concept of tumor development as well. The general concept of tumor development initially had been that probably stem cells in the normal tissue, which would form this normal hierarchy, might acquire a whole series of mutations which would eventually create an oncogenic cell, which would then go on to form the tumor. But with this concept of plasticity in the phenotype of the stem cells, we can think about a situation where, in fact, we may be getting mutations which are pushing these cell populations backwards and forwards, up and down this phenotype, and this, in fact, may be a more likely possibility for the way tumors actually develop because of the much larger number of transit amplifying cells which exist as opposed to stem cells. So the probability of getting mutations in this population is actually higher. What Schaefer and Weinberg had actually argued from that kind of a diagram, that if you actually target your therapy only at the cancer stem cells, you really probably will not be curative, because there will be a pool of cancer cells that can be continuously regenerated from the plastic non-cancer stem cells. And they can re-enter the stem cell state. And they also went on to point out that what this also means, in fact, is that metastasis may not necessarily arise from stem cells which were in the existing pool of cells in the primary tumor, but since it's possible to think of a progenitor or a transit amplifying cell moving out and actually translating back into a stem cell at a distant site. So this is very important in the way we think about how these might develop. So thinking then about the issue of treatment response, one of the things that we need to think about then are various features which could affect the way the cells respond, and we're going to focus primarily on radiation, as I said to you here. There is evidence that stem cells may have increased ability to repair DNA damage, which is one of the major factors associated with cell killing by radiation. There's also evidence that they may have increased levels of free radical scavengers, which will reduce the amount of damage that may be developed. And there are various other features of stem cells that might impact on their ability to respond to radiation. One of these being hypoxia, of course, as we know that hypoxia can modify radiation sensitivity.
So this slide shows data from one of the early studies looking at the question of whether stem cells have increased radioresistance relative to progenitor or transit amplifying cells. What we see in the left panel is the growth of two different glioblastoma cell lines, and you can see they're either CD133 positive or negative sorted cells, and down the left column you see the cells growing without radiation and fairly similar, just plating there. But in the middle, in the second column over in the middle, you see that the CD133 minus cells are showing less growth following a dose of 5 gray, suggesting that those cells are more sensitive than the CD133 positive cells. And you see that's true for both of the two glioblastoma cell lines which were looked at in this study. The other two panels here relate to checkpoint kinase activity, which we're not going to really discuss. In the right panel, what is shown is an alkaline comet assay analysis. For those of you who are not familiar with this analysis, it's an electrophoresis technique which allows one to look at broken DNA. So it's really looking at strand breaks. And because it's alkaline, it's looking at both single and double strand breaks. So if you look at the top panel, you see that what's happened here is that the cells have been lysed and they're put in agarose and then undergo electrophoresis. And in the top panel, you see that without any treatment, you just see the nuclei. But in the second panel down, what you see is the nuclei, but now you see the DNA streaming out to the right, forming a kind of a tail. And you can quantitate the amount of DNA which is pulled out of the nucleus by measuring the concentration of the DNA in the tail and relating it to the amount of DNA which is in the nucleus still. And you see that in the third panel down, the so-called 18-hour panel and the 30-hour panel, you see that mostly that tailing out of the DNA is lost because the strand breaks have been repaired and you don't see as much DNA which is able to get out of the nucleus. And in the quantitation just to the right, you can see that at 1 hour, both the CD133 positive and CD133 negative cells were showing the same amount of DNA really streaming out of the nucleus, but that at the 18-hour and the 30-hour point, the CD133 positive cells in the open bars are actually showing less DNA streaming out, indicating that they're actually repairing the DNA faster. So this suggests then that one of the potential mechanisms by which the cells may be more resistant to radiation is that they have increased ability to repair DNA damage.
So here's another example using again a mammary tumor cell line where we have CD44 positive, CD24 low as the marker, and you can see that here the cells were grown as mammospheres, and if they're grown in culture, you see this with very few cells in this population. But if they're grown as mammospheres, you see a much higher proportion of cells in the CD44 positive, CD24 low population. And they looked at the ability of these cells to form colonies following radiation, and you see that the cells from the mammospheres are more resistant to radiation than the cells from the single cell populations. And they also looked at DNA damage. In this case, they used gamma H2AX, which is a marker of DNA damage. The phosphorylation of the H2AX protein is associated with the presence of double-strand breaks. And what happens is that at early times after radiation, you see the development of these breaks, and you're seeing that here over that 60-minute period. and then over longer time periods, those breaks are repaired. And so what you're seeing here in this case is that the cells which are in the population, which is the single cell population, are actually showing many more breaks at 60 minutes after radiation. These are different doses of radiation here than the cells which were in the mammospheres. And they saw the same thing when they looked at free radicals immediately after radiation. There were more free radicals in the cells, suggesting that these were associated with the DNA breaks. This is the very interesting data set, the only one actually from clinical data which tries to relate stem cell markers to treatment outcome. Here we're looking at head and neck cancers, particularly larynx cancers. And what was done was they looked at CD44 as a marker for stem cells in this population, using immunohistochemistry really to examine the level of CD44 expression in the tumor cells. And they related the level of CD44 expression in the tumor cells to the possibility that the fraction of tumors which would actually recur. So here what we're looking at is the fraction of tumors that didn't recur or did recur in relation to the level of CD44 expression. And you see the higher levels of expression, you tend to see more recurrent tumors, and that's plotted out here, where they're plotting the odds of recurrence versus the CD44 score, showing that the odds of recurrence rises as the CD44 level actually goes up in the different tumors. They went on using cell lines, head and neck cancer cell lines, to really try and ask the question: what is this due to? Is it due to an increase in radiation resistance of the cells which are expressing CD44, or is it just due to an increased fraction of cells which are in that population? And here they're looking at plating efficiency as a measure of the fraction of cells which are in the population which have the ability to grow, and they show that the plating efficiency of those populations rises with the level of CD44 expression. Whereas when they looked at the radiation sensitivity, they really didn't see much change in radiation sensitivity as a relation to CD44 expression. So what this data suggests, in fact, is that this is primarily due to the number of stem cells in the tumors rather than differences in their radiosensitivity. But it is important to recognize that radiation may, in fact, impact on the transition between the non-stem cell and stem cell phenotype. Here is data from work done in mammary tumors, where they looked at 3 different patient samples, and they looked at the percent of cells expressing the aldehyde dehydrogenase marker. And so what was done here was that the patient samples were stained for aldehyde dehydrogenase activity, and then they sorted the aldehyde dehydrogenase negative cells, and then they plated them in culture, and they irradiated them with 2 doses of radiation, either 4 or 8 gray, and then they looked for the fraction of aldehyde positive cells 5 days after radiation. And what you can see is that the irradiation actually resulted in an increase in the fraction of cells which were expressing the aldehyde dehydrogenase marker. And that was true for all 3 of the patient samples that they looked at. And they also looked at a couple of mammary tumor cell lines and saw essentially the same effect. So just as a kind of a summary then, if we think about how cancer stem cells might impact on the tumor control by radiation, there are a number of different factors which are important. As we said, there's the actual total number of CSCs in the tumor. If you have a high number, you would expect more likely to get recurrence. If we are actually driving cells into the stem cell phenotype used by the radiation treatment, then if that happens rapidly, it's a high probability of that happening, and again, you would expect a lower probability of the chance to cure. You would have a high chance of recurrence. Equally, if the intrinsic radiosensitivity of the cell populations is different. If the CSCs are actually more resistant, as we've shown that they are in some cases, again, you would have a higher probability of recurrence. And hypoxia, of course, if there's a lot of hypoxia, that will impact as well. So the overall summary then really is that we can identify stem cells using specific markers, and there's evidence that they may be more resistant to treatment than progenitor and transient amplifying cells, but the extent to which that's true in different populations is really not very well known at the present time. What we know about stem cells is that their phenotype does appear to depend on a microenvironmental niche, and one of these niches may in fact be hypoxic. And that, of course, could affect treatment response, as we've talked about. We also know that this phenotype can be associated with this EMT transition, and therefore that may enhance metastasis in populations that do a lot of this. Very importantly, we know that the stem cell phenotype can be plastic, and the progenitor and transient amplifying cells may actually reacquire stem cell properties. And this is a very important issue in relation to the fact that the treatment might actually impact on this plasticity. And this, of course, effectively changes the number of cells in the population. So it reduces the initial information that would be useful potentially. So just wanted to acknowledge that many people in Toronto are working on the hypoxia issues, and these are listed here. The two people who did the work with the cervix cancer xenografts are Naz and Tamara, and certainly like to thank them for all the work that they have done. And I would like to thank you for listening, and I'd be happy to address any questions. Thank you.
All right. Well, thank you, Dr. Hill. So let's go ahead to the question and answer segment. So if you haven't yet submitted a question, now is the time to do so by clicking on the Ask a Question box at the bottom of your screen. So let's go ahead and see what questions have come in so far. So the first one here, it says, in our hands, we can get fibroblasts to form spheres in spheroid media. How specific is the sphere-forming assay for cancer stem cells?
So I think the answer to that question is really unclear. Certainly, if you take tumor cells and you grow them as spheroids, it appears that you can enhance stem cells, but it's clear that you can grow other cell populations in the spheroid environment. So it's not specific to you don't get a specific situation when you grow cells as spheroids. So there's certainly lots of data which suggests that other cell populations can be grown as spheroids.
Okay, on to the next question. It says, what are the main differences between a so-called cancer stem cell and a slow-growing cancer cell with a different genetic mutation than the dominant cancer cell population within a tumor?
Well, it's not, I don't think, a distinction that one can really make in this context. Different tumors clearly contain different clonal populations, if you like, within the tumor, and I think most of the evidence is that these clonal populations probably all contain at least some stem cells. So the concept that there is one dominant clone at any one time seems to have some value, but it's clear that there are other populations in there, and there are probably stem cells within those populations as well. So it's not really a distinction, I think, that we can easily make.
Okay, on to the next question. It says, you mentioned the issue of stem cell plasticity. Can you expand on the idea that stem cell plasticity may relate to the development of metastasis?
So there are two issues here, really, which we can think about. One is the issue of the possibility that the stem cell phenotype may be associated with the so-called epithelial to mesenchymal transition. And it has been thought that phenotype actually predisposes cells to be able to form metastasis. On the other side of that question is the issue that we talked about at the end about whether, in fact, it's necessary for a cell to be a stem cell at the time that it actually escapes from the primary tumor in order to be able to form a metastasis. Because if there is this possibility of plasticity, then maybe that type of effect can occur at a metastatic site. So we can think about the possibility that a tumor might have not have stem cell properties, but escape from the tumor and move to another site in the body, and then at some later time redevelop a stem cell phenotype for reasons which we don't really understand at the present time, and then grow to form a tumor. And this might certainly explain why one sees late metastasis in some cases, although that's purely speculation at the present time.
All right, next question. What do you think of the studies on the hyperbaric oxygenation to enhance chemoradiotherapy, such as publications of Koshi on glioblastoma?
Yeah, there's a long history of the use of hyperbaric oxygen in radiotherapy that dates right back to the late 1950s, actually, where they were focusing on trying to oxygenate hypoxic cells in the tumor, and it was in fact shown that in those studies that you could enhance treatment response using hyperbaric oxygen, putting patients into hyperbaric chambers. Turned out to be a very difficult thing to do, in fact, and there were a number of accidents which resulted primarily in mostly this falling by the wayside is really a technology that couldn't be easily implemented in the context of radiotherapy. But certainly it was shown early, as I say, to be a technique which could be potentially beneficial. Nowadays, we might also argue about whether increasing the oxygenation might actually reduce the stem cell proportion, and that might be part of the reason why it was effective. But I don't think that we have any clear evidence at the present time that which way that's going, because virtually all tumors do contain at least some hypoxia, and that can be measured. So one could certainly set up an experiment where you could actually answer those, certainly address those types of questions.
All right, on to the next question. Simply, why do tumor cells prefer hypoxic conditions?
I'm not sure that that's a correct interpretation. I mean, hypoxia develops in tumors, as far as we understand it, because of the problems associated with the development of a good vasculature. And so the vasculature in tumors tends to be very poorly organized, and as a result of that, you get regions where the cells do not have good access to oxygen, and cells have ways of dealing with that problem. I mean, I think I don't know whether that, and we know that in the embryo, for example, there are certainly are periods of time when cells have to deal with low oxygen levels, and it may well be that cells are kind of programmed to deal with hypoxia for various reasons, and they're able to bring to bear these kinds of responses when there isn't a suitable oxygen supply. And we see that in tumors largely because the vascular structure of tumors is generally poorly organized.
Okay, on to the next question. It says, Broxmeyer's group last year had a very important paper in Cell showing that hematopoietic stem cells, if exposed to room air during isolation, suffer a tremendous loss of self-regenerating cells due to hypoxic shock. Have you done any work in which you protected cancer stem cells from room air oxygen during isolation?
So the answer to that question is no. We haven't done any work of that type. I assume what we're actually talking about is oxic shock rather than hypoxic shock. But I think that certainly is a very interesting observation, and would not be too inconsistent with the concept that that cells respond. They change their metabolism when they're under low oxygen conditions, and certainly cells inside the body or in tissues, except maybe in the lung, are never really exposed to the kind of level of oxygen which we see in room air. So there's a shock associated with that process always, and it may well be that the cells have to adapt to that. And so when you take cells into culture and grow them under normal air conditions, we know that they're adapting and changing, and this may be one of the issues that they have to adapt to.
Okay, I think we have time for a few more questions. The next one is, the studies with the cervix cancer PDX model that you described suggested that many of the cells expressing a stem cell phenotype were hypoxic. Are you able to relate this observation to a treatment response?
Well, we certainly have been looking at the response of a number of these models to radiation treatment, but we haven't at this time got enough data to really relate the issue to the numbers of stem cells that we think exist in these different cell populations. So I don't have anything kind of really erudite to say in that case. I mean, certainly we would anticipate that if the cells are in hypoxic environments and most of the stem cells sit in those environments in these tumors, then they would be more resistant to radiation. But important, I think, to remember what I told you about different oxygen concentrations. We're using CA9 as a marker for hypoxia, which is turned on by essentially by HIF1 primarily. So we're looking at cells which are generally at higher oxygen concentrations than we would be if we were using some of the other hypoxia markers. So these cells will not be fully resistant to radiation. Well, cells are not fully resistant to radiation due to hypoxia; they just become more resistant. But they won't be as resistant as they could be. They're some kind of intermediate between the oxic state and the hypoxic state in this particular regard, probably.
All right. We're technically up at 1 hour mark here, but there's a lot of great questions, so I'd like to go just a few minutes over for whoever would like to stay to take as many questions here as we can. So the next one is, what is, in your opinion, the role of mitochondria in CSC metabolism?
I don't think I have anything useful to say there, really. I mean, mitochondria obviously plays a very important role in the metabolism of oxygen, and certainly in the context of cancer cells, we know that there are changes in metabolism related to the so-called Warburg effect, where cells use more glucose and glucosamine to actually, not glucosamine, sorry, but they use more glucose to actually generate energy. But the role of mitochondria there is I don't think is certainly, I do not have anything which I can really tell you about how that relates to stemness, and I'm not aware of any data where people have looked specifically at that issue.
Okay, we can go to the next question then. This one asks, is cancer stem cell differentiation rate the same as normal stem cells?
Again, I don't think there's any real information on that issue. I don't know of any really good data which would really argue that way. I mean, if we think about differentiation is a process which occurs after in a division process, then the rate of division might impact on that kind of information, and so the proliferation rate of different cell populations, of course, is very different in the body. In the blood, there may be a relatively high proliferation rate of certainly of the transit amplifying cell populations, although the stem cells may in fact be mostly quiescent and only a few of them divide at any one time. Whereas in tumors, maybe there's a higher proliferation rate, that's pure speculation, of the stem cell populations. But again, there's general thought that they maybe tend to be more quiescent than the other populations. So it's not, I don't think, an area where we really have a lot of good data at the present time.
Okay, I think we have time for one more question, and this one here actually has 2 or maybe 3 parts to it. So I'll read it all out, and maybe we'll have to go back and tackle it bit by bit. It says, in our team, we are looking for CSCs from glioblastoma and melanoma. In our experience, common markers such as CD133 and P75 do not work to identify this cell compartment. Do you have any advice on what to look for more specific identification? It also says, we are currently working with hypoxia for CSC enrichment, but this stress also induces autophagy. Do you have any ideas about the connections between both processes?
So, talking about the first issue, certainly I think that you're not alone in having problems with kind of identifying suitable markers, and I guess one needs to recognize, as we said earlier, that this varies from one tumor population to another. So in one tumor population, you may get quite nice CD133 expression, for example, and this may give you an enrichment for the stem cell population. In another tumor, you may not see this kind of nice relationship, and I think that this is not an uncommon experience with people who are working with stem cells, although this kind of information maybe doesn't kind of get published as often as the ones that show clear markers do. So probably it may be helpful to work on the populations where you do see these kinds of effects with a marker, but you can certainly look at different markers. There are a number of different markers out there for different populations, and it may be that you can actually find another marker which will actually help you better with the identification of the populations. I showed you data set for breast cancer, for example, where it looked like there might be two populations of different markers, or both of which had stem cell phenotypes. So this is certainly something that one needs to be aware of. It's not an easy situation, and it's not an all or none phenomenon in all tumors. So the same marker doesn't necessarily work in all glioblastomas, and that might be part of the problem. Going on to the issue of hypoxia and autophagy, that's certainly been demonstrated that hypoxia can in fact push cells towards autophagy, and this obviously relates to the issue of needing to actually survive under conditions where you don't have access to resources to produce the energy you need to survive. So autophagy is a process which cells can use to help them survive conditions which are stressful, but it also is a process which can lead to cell death. So both of these things can happen, and it depends a little bit on the oxygen concentration that you're working at. So the point that we were making before is that different things happen at different oxygen concentrations, and so this may depend on the conditions that you actually have in the culture as to what kind of results you're getting. Bear in mind that the cell density that you're growing the cells at, and the thickness of the medium over the culture, if you're growing them in culture, can also all affect the oxygen concentration that the cells actually see, even when you've got a given concentration of oxygen above the medium. So these might be factors that you need to have under tight control in order to get good control of what level of oxygen you're working at and what's happening under those conditions.
All right. Well, we are now out of time, so I'm going to conclude the question and answer session. Today's webinar has been recorded and will be available for viewing in the next few days. We will send you an email with details on how to access the recorded webinar, along with instructions on how to personalize and print a certificate of attendance, as well as download a PDF of the slides. So on behalf of today's speaker, Dr. Richard Hill, and our sponsor, Baker Ruskin, we sincerely appreciate you attending today's webinar and look forward to your attendance at future events from Current Protocols.